The overall goal of this methodology is to assess both the adhesive properties and cellular biocompatibility of the proposed hydrogel composite for intervertebral disc tissue engineering of the nucleus pulposus.
This method can help answer key questions in the field of tissue engineering, like how to design and prepare thermoreversible hydrogels with bioadhesive properties for cell encapsulation. The main advantage of this technique is that we are able to demonstrate in vitro studies with an injectable, thermally sensitive composite to evaluate its use as a potential replacement for the nucleus pulposus.
In this current study, we evaluate a novel hydrogel scaffold by performing both tensile mechanical tests and cellular viability assays.
Demonstrating the biocompatibility assays will be Emily Schmidt, Mark Dittmer, Edward Goldschmidt, and Francesca Giginis, students from the departments of Biological Sciences and Biochemistry.
Prior to the synthesis of the bioadhesive hydrogel, purify NIPAAM monomer and MCS as described in the text protocol. Then, co-dissolve 10 grams of purified NIPAAM and 2.209 grams of MCS in 232 milliliters of deionized water. Purge the solution of oxygen by using inert nitrogen gas. Maintain a vigorous yet low gas flow rate to prevent the solution from bubbling over for 15 minutes. Continue purging the solution with nitrogen gas and add 0.976 milliliters of TEMED. Next, initiate the polymerization reaction by mixing 97.6 milligrams of ammonium persulfate. Mix the solution for approximately 20 seconds.